Hello everyone and welcome back to the week 11 of NPTEL course in pharmacognosy and phytochemistry. In this week as well, we are seeing different methods of quality control. Now in this session, we will see a slightly different method of quality control, and that is biological method. Okay. Before we go to the biological method, just a quick recap. Till now we have done organoleptic method, microscopic, macroscopic, physical, chemical methods of analysis. All of these methods help us ascertain the quality, that is the identity, purity as well as the content of the herbal drug material. Now what are these biological methods? Whenever we say biological methods, we take into account biology. That means we are going to use living organisms either in form of organism as whole or the enzymes or the products or certain cells purified from those organisms. That means we are going to perform a method of analysis, that is a quality control method, by using biological specimen or sample or enzymes or even in certain cases the fluids. So let us see the different biological methods that can be used for herbal quality control. So biological methods, mostly what we are discussing are quantitative methods. In this session we are going to discuss methods such as hemolysis test, bitterness value, microbial contamination and efficacy studies. Now efficacy studies is a large domain. So efficacy studies means when I am consuming a drug, is it really going to be showing its activity as promised? Now this can be done by actually administering the drug to certain individuals or in some cases to the lab animals or experimental animals more specifically, or in some cases it can be observed by expression or suppression of certain proteins and enzymes in cells which are grown ex vivo. So efficacy studies is going to tell you whether your medicine is going to work or not. It's a big and a very broad area, and efficacy studies varies as per the therapeutic value intended. So if I have to study for anti-diabetic effect, I'll have different kind of assays. If I have to do anti-cancer effect, I'll have a different kind of assays. So efficacy studies being a little bigger domain, we will not be covering in this course, but in today's session we will be covering more specifically the hemolysis test, bitterness value, and how do you assess microbial contamination in given sample. So let us start with the first sample, and that is your hemolysis test. Now hemolysis, as the name indicate, somewhere you are going to take this blood and you are going to hemolyze it. That means you are going to cause the lysis of your RBC. Now why does this happen? So there are certain compounds in plants that are called as saponins. Now saponins have a good ability to play with permeability of your RBC membrane, thereafter disturbing it and kind of eventually rupturing it. This happens by binding to different locations, and the eventual product is the heme, which is trapped inside the RBC, is released. Now when this heme is released, your plasma becomes colored because of the presence of heme, and that color you are going to see. So for that we will require few things, and what we require in terms of biological sample is going to be your RBC suspension. Now when we say hemolysis, you want to release the hemoglobin out of your RBC. So you are going to require RBC. So from where do you procure this RBC? This RBCs can be procured from rabbit. In some cases even mice are used, but not clearly for this specific method. But bovine RBCs, in some cases even human RBCs, if at all they are available. Now in order to procure this RBCs, you procure blood first. Okay. So you draw blood out of animals, bovine animals, humans, and this blood you're going to separate it component. You're going to separate the plasma, you're going to separate the WBC, RBC and so on. This is done by centrifugating the blood at a very high speed or at a very high RPM, wherein your cells settle based on their density. So you will see a centrifuge tube, and in this centrifuge tube, after centrifugation, the top layer will be a very clear plasma. After plasma, you will see the little less denser WBC and platelets. And because of the heme, the bottommost plug or that bottommost layer is actually the RBC. So you have different cells settling at different rate due to change in their weight, and the bottommost layer is your RBC. So this, what is done is, this upper layers are carefully removed off, and this RBC plug, what is there, is resuspended, preferably in an anticoagulant initially till the plasma is there, and later on using your phosphate buffer, basically a saline phosphate buffer, because isotonicity here is very important. If you do not keep your solutions isotonicity, because they will become if the solution becomes hypertonic or hypotonic, they might shrink or rupture. We do not want the cells, or especially our RBC cells, to rupture or shrink, and as a result we keep it in a isotonic saline buffer solution suspended. So generally what you prepare in a lab is somewhere between 1 to 5% RBC suspension in a solution of your saline phosphate buffer. Now you take this, what you will see is, again if you recentrifuge it, again your RBCs will settle. Now this time what you are going to do is, you are going to add your RBCs to your test tubes containing different solution. Now you cannot keep a really big test tube, so imagine a very small test tube, and in each of it we have just added 0.1 ml of your RBC suspension. Now in addition to that, you will add your extract. So in this case, say for example, I add here 0.1 ml extract, 0.2 ml extract, 0.3 ml extract and 0.4 ml extract. Now if at all it contains saponins, what is going to happen is, as the concentration increases, it is going to cause lysis of large number of RBCs. So more the amount of saponins, more the lysis will happen. And what is the product of lysis? The product of lysis is heme is going to escape into this saline buffer. So initially you will see a very clear or very little hemolysis, but as the concentration of saponin increases, it becomes increasingly red. That means more and more of this heme is released into the saline phosphate buffer. Now what you do is, in order to quantify this, you can use colorimetry. So just measure the optical density of this solution at about 630 nanometers, and then use a standard saponin, of which you know what is the LD30 or especially HD, hemolysis 50 value. Now HD 50 value is a value of the standard saponin that causes 50% of the RBCs to lyse. Now you will see that, and whenever your value matches to that, you can say that this is the concentration, which is the HD 50. Now you compare it versus standard saponin, you already know the concentration of standard saponin. So this is the concentration of standard saponin, say 100 microgram per ml causes hemolysis of 50% of your drug. Now check the extract, say your extract will also contain certain things. So say for example, you will say about 400 microliter causes lysis of about say I am just putting it 400 microgram per ml causes HD50. Now what does this mean? This means that you require 4 times more concentration of your test sample as compared to standard sample. So if this has 1 mg, So this has 0.25 mg of your saponin. So this is how this method is used for quantification of saponin. Now we go to the next method. The next method is a little obsolete now because of machine learning and electronic tongue along with HPLC, but this is a method which was earlier used, and that is your bitterness value. Now what is bitterness value? Bitterness value is the amount of bitterness that is felt after you consume, say for example, dissolving 1 gram of quinine in 2 liters of water. So you take 1 gram of quinine, dissolve it in 2 liters of water, especially the salt, quinine hydrochloride is used, and then when you see it effectively, this is the concentration that you reach, 0.5 mg per ml. Now that is considered to be a bitterness value of about 2 lakh units. Now compared to that, what dilution of your sample, say for example there are numerous bitters, strychnine, brucine, or even emitin, even your andrographolide, or even we saw your iridoid, swertiamarin. So you are going to take these solutions and you are going to taste in a similar manner. So you are going to dilute it till you reach a threshold bitterness value, and then you check what is going to be as equal to the same dilution or same bitterness as that of quinine diluted in 2 liters. Now this can be given by a formula 2000 by A, by 2000 by A is kind of reciprocal, 1 gram of quinine in 2000 liters. So how much is your sample? So A is actually the bitterness value is actually the reciprocal of the concentration. So you need to take it ulta, and that is exactly the reciprocal value of the concentration needed to match the bitterness of 1 gram of your quinine. Now once this is done, you put your A value and then you will get your bitterness value of the sample. Now moving on to the next one, but a very important one, and that is determination of microbial contamination. Now when you see your synthetic APIs, most of them have been synthesized by using chemical methods, and as a result they are not food for the microorganisms. Many of the chemicals don't really harbor microbial contamination, but when you come to herbal drugs, they are all natural, they are carbohydrate rich, they retain certain amount of moisture, and as a result they are more susceptible to microbial contamination. In that case, it is very important to check the microbial contamination values, especially for your herbal drugs. Now one important difference is microbial contamination essentially is different from sterility. Sterility is referred to a term when you say complete absence of microorganisms, whereas determination of microbial contamination, sterility is something where you say it's complete lack or absence of microorganism, whereas here you will say that you're going to quantify how much microorganisms are there. The reason for this is most of the herbal drugs we consume via oral route, and as a result we have our stomach acids and we have our immunity which will support in our defense function. So as a result, these drugs or these herbs need not be sterile. But in some cases, if the microbial load is too much, imagine you trying to eat up the fungus. What is going to happen is somehow or the other you are bound to get the infection. So there is a particular limit beyond which the microbial contamination should not be there. Your product need not be sterile, but your microbial contamination should be curtailed is a proper word, and that is why we do the determination of microbial contamination. Now determination of microbial contamination has two important aspects. First thing, we are going to see how many alive or viable microorganisms are surviving in it, and that is referred to as your TVAC, or often called as your total viable aerobic count. The reason we do total viable aerobic count is most of the herbs are kept exposed to the atmosphere, and as a result majority of the microorganisms that survive on these herbs are all aerobic. But yes, there is a chance that certain anaerobic microorganisms also do sustain, but they are not kind of obligatory anaerobes. So total viable aerobic count will give you a rough estimate of how much contamination your drug carries. Now how are you going to do, or how are you going to measure the microbes in the herb? Now in order to do this, you require something called as a media. Now this media will essentially help you in growth of microorganisms as well as enumeration of microorganisms. Now the growth of microorganisms and enumeration of microorganisms can be done in certain nutrient rich media, often referred to as your nutrient broth. So this nutrient broth kind of becomes home to that microorganism. They kind of multiply in that and gives rise to colonies. These colonies can then be counted in case of a plate method, or this growth can be counted as increase in turbidity. More microorganisms, the difficult it will be for light to pass it. The solution will lose its clarity. Imagine fungus growing in water. So what is going to happen? Initially it is clear, now it is not. So there is a change in optical density, and that is also one way to do it. Now we come to the second part. The second part is pathogenic microorganisms. Now these are the microorganisms which definitely are going to be injurious to us if we consume it, especially if the patients consume it. Already their health is compromised, and in that case these microorganisms, especially the opportunistic ones, what will they do is they will infect the patient and give rise to secondary disease. Say for example Salmonella typhi can infect, and then you will get typhoid in addition to the disease you are already suffering from. So we check this opportunistic but pathogenic microorganisms using again plate method, as well as nowadays we have certain methods such as your polymerase chain reaction with ELISA, which helps you detect these microorganisms even with a very lesser concentration. So let us see this method, what is total viable aerobic count. So in this case we generally take, or most of the pharmacopoeias generally advise you to take about 1 gram of drug. Now this 1 gram of drug that you are going to take, you are just going to take in atmosphere, and it is bound to be contaminated with microorganisms. Now this 1 gram of drug you are going to transfer to a flask containing sterile media. So if you remember, maybe in your microbiology or biochemistry you have this conical flask with a non-absorbent cotton. Now this should contain your sterile media, and in that you are going to add your 1 gram of sample. Now when you add your 1 gram of sample, you are going to shake it thoroughly. Why? Because whatever microorganisms are there in your sample should get transferred to the media. Now if the sample is very dense, you allow it to settle, or in some cases even a coarse filtration with the sterile filter paper is recommended. Now but that leads to what is called as inaccuracy. So in some cases it is filtered, in some cases it is centrifuge. So after centrifugation you get a clear supernatant, and that supernatant is used. Now again depending upon the conditions, in some cases where they are more of lipophilic drugs, even surfactants to a little concentration of 0.1% may be added to facilitate the transfer of microorganisms to the media. Now once you have the sterile media, you kind of do what is called as a serial dilution. Now serial dilution, you already have 1 gram and say for example 100 ml in media. Okay. So your concentration becomes 1%. Okay. Let's put it as 1%. Okay. Now this, if you take 1 ml of it and dilute it 10 times with just water or another sterile media, the concentration will become 0.1%. Now again dilute 1 ml of this, make the volume to 10 ml, you will get 0.01%, and then finally again take 1 ml of this, add around 9 ml of sterile media, and you will get 0.001%. Now why is this dilution important is, you can see there are certain microorganisms, and from the samples you cannot really make out visually by seeing it what is the concentration. So say for example, if I have to plate it, that is if I transfer this to a media, say 1 ml here of this, 1 ml here of this, 1 ml here of this. So when I say my solution is concentrated, you see there is a film like structure being formed. In that case, it becomes difficult for me to enumerate how many microorganisms are present. I further dilute it. Now at least I am seeing some colonies. Now what happens in your microbiology, every microorganism which is kind of suspended in your plate, initially by a pour plate method or spread plate method, you will see that gradually gives rise to one colony. So one colony initially was just one microorganism. So these are called as colony forming units. So each CFU is considered to be one microorganism. Now if you see the last dilution, this is the one I want because I can easily count how many are there. So if I say my 0.001 gram contains 4 colonies or 4 CFU to be specific, my 1 gram contains how much? So I will just multiply it and I will get 400 CFU, okay, 4000 CFU. So in this case, what do I get is, if I say their limit is, say for example, my drug should not contain more than 1000 CFU. So if it is 1 gram should not contain more than 1000 CFU, and if my reading is going higher than 1000 CFU, say for example 4000, that means my sample is failing the microbial contamination test. But on the other hand, my limit says 1 gram should not contain more than 1000 CFU. If at all due to some reason my CFU is just 400, that means it is less than 1000 CFU per gram. So what do I get in that case? So what I get in that case is, it is passing the test because 1 gram contains less than 1000 CFU. So I can do that. Now in this case, I can also selectively bifurcate between the bacterial count and the fungal count. For my bacterial count, this is a little non-specific, which also allows certain fungi to grow. I can use my soya bean casein digest media and I can incubate it at 37 degrees Celsius for a day and I will get my colonies. Whereas for fungus, I can use something more specific, which is your Sabron's dextrose agar, which is slightly at an acidic pH, which does not allow the bacteria to grow, and this can be incubated. Fungus is preferentially grown at a slightly lower temperature. So mostly you will see it incubated between 20 to 25 degrees Celsius. I can observe the same thing, but when it comes to the growth for fungus, it is more of a filamentous kind of growth which is seen. One other method is also, I can do it or grow it in a liquid media referred to as broth. In that case, I will see the turbidity. More the microorganisms, more will be the turbidity. So I measured light scattered by the organisms at about 600 nanometers, and based on how much light is being transmitted, I can take a call whether the microorganism count is high or it is low by comparing it with a standard optical density values. Now coming on or moving on to the specific pathogenic microorganisms. Now pharmacopoeias or WHO says, say for example acacia, 1 gram of acacia should not have more than 1000 CFUs, and we just saw we can do that. But when it comes to pathogenic organisms, the pathogenic organisms are something which we do not even require 1 CFU to be there. 1 gram should not even have 1 CFU. In certain cases or in very rare cases, they do allow up to CFU, 10 CFU per gram, but that is not always the case. So pathogenic microorganisms are the microorganisms we want to get rid of. which we do not want to have in our herbal drugs, and as a result a good quality control method should selectively check if these microorganisms are present and to what extent they are present. So we can do that by harvesting, or we can do that by allowing this dilutions to be poured on selective medias, which will allow us to check only for the presence of pathogenic microorganisms. So the selective medias are medias which will allow more of a pathogenic microorganisms to be alive, will kill all the normal or other non-pathogenic microorganisms, or in certain cases it will still allow the other non-pathogenic microorganisms to grow, but they will show a different color as compared to your pathogenic. So there might be a color difference, or there might be a selectivity in growth, and such media are called as selective media. So for E. coli, more selective media are your sorbitol McConkey media. Now this is more for your what you call it as lactic acid digesting bacteria. So in this case, whenever you have your sorbitol McConkey or eosin methylene blue agar plates, they have a typical indicator, say something like a phenol red. So what is going to happen is, first preferably your lactose will be digested to lactic acid, or fermentations will happen and more of this acids will be produced. As more and more of this acid is produced, say for example by your E. coli, you will see that your phenol red will make it red, and as a result your E. coli colonies will appear red, whereas other colonies might appear just white because they are not producing any acids. Same applies with your eosin methylene blue, wherein your media causes acidity wherever your E. coli is growing, and that gives a little green metallic sheen. Now coming to salmonella, salmonella specifically digests xylose, then it kind of digests your lysine also. Now after this what happens is, you will see this salmonella kind of reacts with certain metals like your iron in this, or in certain cases even lead, and that gives rise to what are called as black centers or black colonies. So you will see a black centers or black colonies coming out of precipitation due to reaction with metal ions. When it comes to Shigella, you have Salmonella Shigella media, which is non-specific for both, that is Salmonella and both Shigella will grow. Again that gives you a black color colonies, or specifically Shigella broth, which uses a tryptic soy broth with yeast extract. Now coming to Pseudomonas aeruginosa is more often a organism which is known to cause blindness. Now this can be detected selectively. The agar used for this is cetrimide, that is your cetyl trimethyl ammonium bromide agar, and what happens is very few organisms can stay or survive on this, and as a result not only will the Pseudomonas stay on it, it starts producing different pigments. These pigments include your pyocyanin and pyoverdin. Now this pyocyanin and pyoverdin gives you a little blue, green and yellow colonies, and as a result wherever the Pseudomonas is growing, the media or the plate is seen with greenish or greenish yellow pigments. So presence of this pigment itself in your cetrimide agar is an indicative that your Pseudomonas aeruginosa is present. The next one is the pneumonia causing Klebsiella pneumoniae, which is selective media for this is your chrome agar. Now chrome agar also contains your indicators or your pH change indicators, and this gives you metallic blue color colonies on reaction with certain indicators. So these are more selective medias. This will help us tell. Now in certain cases you will observe that when you see a total viable aerobic count, the incubation time is very short, maybe just 24 hours. But when you have to grow the pathogenic microorganisms, the pathogenic microorganisms are not commonly occurring organisms. So their concentration even in herbal sample is pretty less, but still you have to ensure that they are absent. In such cases, these herbal samples are taken and first grown in an enrichment media. this enrichment media will allow selective growth of this pathogenic organism. So, all these organisms will be first grown, or the samples of which will be first treated with enrichment media for a day of 2. So, initially for a day or 2, when the enrichment media is used, you will see, preferably your E. coli will stay, rest all will die. Preferably your salmonella will stay, rest all will die. So, such selective enrichment medias are used, and once the microorganisms are enriched, then they are transferred to this plate. So, comparatively, even for the growth, the incubation time lasts somewhere between 24 to 48, and some cases goes to 72 hours, because their count is very less, and in order to make a good estimate whether the microorganism is present or not, it is very vital to see the end results. Okay. So, here are few references which you can refer to in case where you want to check your microbial growth, microbial media, your hemolysis, or different biological methods. Like I said, efficacy testing is still there, but since efficacy testing is a bigger volume to handle, it alone is a big subject. Considering or taking into account different disease require different animal models to prove its efficacy, we will restrict ourselves to this biological methods. So, thank you everyone for your patient listening.